So this week we are going to be taking what we've learned in genetics and applying it and understanding it and using it. And one of the ways that we do that is using the statistical analysis called the chi-square. In a nutshell, what the chi-square tells us is, do our data, the data that we are collecting, fit what we expected them to be? It tells us if the difference between what we expected in our data and what we actually observed in our data is too much. Is it due just to chance? Or is it actually telling us, hey, you know what? Your hypothesis is unsupported or falsified. And that's what the chi-square allows us to do. So let's use an example, a simple example from genetics that we've been learning. Let's use purple and white flowered peas. You've heard too much about this in your genetics lectures, so this should be really, really familiar. All right, let's take a true breeding purple, which tells us that it's homozygous dominant. For the flower color, and a true breeding white flowered plant. And that tells us that it's homozygous recessive. And all of this is based on the genetic analyses that have already been done. So it's been predetermined that purple is dominant to white when it comes to flower color in peas. So in order for an individual to have white flowers, it has to be homozygous recessive. But true breeding in both of these cases tells us that that individual is homozygous. So we need to know it's a true breeding purple to know it's homozygous dominant. If we breed these, so let's take this one. All right, this one is going to be big P, big P. This one is going to be little p, little p. If we breed them, big P times little p, little p. We first will make some gametes. So let's have this one be the mom. We'll give her some eggs. She really can only make big P eggs. Let's just do 2 because it's convenient. Let's say this is the dad. We'll make him some sperm. Little p, little p. Now if we put these on a Punnett square, we get all heterozygous offspring. So all are purple flowered and all are heterozygous. And what that also tells us is that all are monohybrids. Now, if you haven't read the lab closely, do so, right? This is your chance to go back and be like, wait a minute, monohybrids, what? Stop the tape or stop the video, go back and watch that. So this is a monohybrid, one gene, hybridized, it has one of each allele. So that's what monohybrid means. So we would expect, if we crossed these, 100% of them to be purple. That's not all that exciting. So let's take these and let's do something we wouldn't really want to do with people. Let's make the siblings mate. So we have a big P, little p times a big P, little p. And this is called a monohybrid cross because we're crossing monohybrids. That's all that is. This one, we'll put mom on the left again. Egg, egg. So 50% of her eggs are going to be big P. 50% of her eggs are going to be little p. Let's have this be dad. We'll make his sperm a little bigger so I can write in them more easily. 50% will be big P, 50% will be little p, because he can either do a big P or a little p. Now let's do a Punnett square with this one. Actually, let's just put it here. All right, dad's there, mom's here. Well, you will have big P, big P, little p, big P, little p, and little p, little p. So if we were to do a monohybrid cross, we would expect 75% to be purple and 25% to be white. So 1, 2, 3, these are purple. Even though they have different genotypes, they're still purple. So looking at them with the unaided eye, we would just see purple. We wouldn't know by the fact that they were purple whether they were homozygous dominant or heterozygous. And we have 1 out of the 4 is 25%. Now this is the probability. So every time you look at an offspring of these individuals, when they cross, there's a 25% chance that offspring is going to be white. There's a 75% chance that offspring is going to be purple. So this is all about probability. This does not mean that if you took 4 corn kernels in your hand, you would expect 3 of them to be purple and 1 of them to be white. It just means for each individual that is produced by the cross between these parents, it has a 75% chance of being purple and a 25% chance of being white. Now this is going to be our phenotype. And then we could look at genotype. So if we were to look at the genotype, we would expect 25% big P or homozygous dominant, 50% heterozygous big P, little p, and 25% little p or homozygous recessive. So we could use this if we were to cross these two as our expected. We would expect a ratio of 3 to 1 purple to white. So if we were to count 100 seeds that were the product of these two individuals mating a monohybrid cross, with that 100 seeds, we would expect approximately 75 purple seeds and 25 white seeds. So if you had a little more than 75 or a little less than 75, what the chi-square allows us to do is tell whether those data are close enough, right? Are those data close enough to still support our hypothesis that this is a monohybrid cross, that purple is dominant to white? Are they close enough or are they not close enough? And if they're not close enough, so if it does not fit your hypothesis, then your hypothesis is questionable, is probably wrong. So it should be examined closely. So maybe if your numbers don't fit, if you didn't have the equivalent of 75% purple and 25% white, or if it wasn't close enough to that, if the chi-square tells you it's too far away, your data have deviated too much from the expected to be just chance, that means that you need to look at your hypothesis and see maybe if you were wrong, right? Maybe white was dominant to purple. Maybe this wasn't simple Mendelian genetics. So the chi-square test allows us to determine if any variation you have from what you expected is too much. Right? Too much is a red flag. Is the difference just random or is it statistically significant? Right? Once you do this type of a test, when you use the word significant in everyday language, you're going to stop and think for a second. Now, do I mean significant like important? Or do I mean significant like statistically significant? So we're looking to see if our differences between our expected data and our observed data are statistically significant and will make us question our hypothesis. So for biology, we use a probability or a p-value of 0.05, which is 5%. Actually, there shouldn't be a percent there. 0.05. So basically, if your p-value is equal to 0.05 or higher, then there's a greater than 5% probability that the deviation was random, not significant. Hypothesis accepted or supported. I'm going to go with supported. I like that better. I know that sounds kind of lame. You're like, there's a greater than 5% probability that the deviation was random. So you're 5% sure that your hypothesis is supported, and that's good enough for biology. If your p-value is 0.05 or greater, we accept your hypothesis, it's supported, your data fit, your expected data, congratulations, you can move forward. If not, something else is going on. Right, so if it doesn't fit, if your p-value is less than 0.05, then your hypothesis is falsified. All right, what that means is maybe it's not, so here are some examples. It's not simple dominance and recessive. So maybe it's incomplete dominance. Maybe it's codominance. Or, I need to be able to spell. There we go. Or maybe it's polygenic inheritance. There's more than one gene. Or maybe there's an environmental effect. Had to remember. Okay, effect and effect. So if your hypothesis is falsified, there's something else going on. Maybe it's not simple dominance or recessiveness. Maybe it's codominant, maybe it's incomplete dominance, maybe it's polygenic inheritance. So there are multiple genes involved, like we see in things like hair color and skin color and all sorts of, and height, I think. Maybe it has an environmental effect, like we saw with the hydrangeas in the lecture videos, where in one soil pH they're pink and in the other soil pH they're blue. So you need to think about what might be going on. The other thing is maybe you just have your dominant and recessive backwards. Right, so these are all things to think about if your data do not support your hypothesis. So if your data falsify your hypothesis. This is what a chi-square test allows us to do. So here are some terms you need to know in order to successfully navigate the chi-square test. 1, categories. Keeping in mind that the chi-square test was not invented for biology, so we can use this for all sorts of things. Your categories are your groups into which your data fall. We're going to use an example where we have heads or tails from a coin. Right, in genetics problems, this is going to be your phenotypes or even your genotypes, depending on how you set up your chi-square analysis. Your observed number. This is what you actually see. So the numbers that go into this column are going to be what you actually observe in real life. Let me see if I have one here with me. I don't. But you will be looking at corn. You're going to look at something like the color of the kernels, for instance, or the texture of the kernels. What did you observe? The next one is the expected number. This is what you expected for the numbers for each one of those categories based on the expected ratio. Right, the expected ratio column, this is where you decide, okay, what do I expect? All right, if this is a monohybrid cross, this is how I think. I close my eyes, I see it in my head. All right, in a monohybrid cross, I would expect 3 dominant phenotypes to every 1 recessive phenotype. 3-to-1 ratio. So depending on what you're looking at, you want to think about what you expect the ratio to be. A lot of times I will start here because this is the easiest part for me. Right? Start where you want to. I'm just giving you some tips. Deviation. Right? Your deviation is your observed minus your expected. Just do simple subtraction. And then deviation squared. Right? So deviation is a little lowercase d. D squared or deviation squared. This is just a squared deviation. And then we have your d squared divided by your expected number. Right, so get these values, you're going to divide your squared deviation by the expected number for that category. And then your chi-square value. To get this value, you sum up all the d squared divided by expected values. That's your chi-square value. So you're going to predict a certain number of corn kernels with each characteristic or trait based on this. These seeds are the F2 from a monohybrid or a dihybrid cross. So what we have done was we have crossed these corn plants, and then we have taken the offspring and crossed them again. So we are expecting that this is either a monohybrid or a dihybrid cross, the results of that, depending on which part of the lab you're talking about. The seeds that you are going to be looking at in lab are taken from a single ear of corn from this cross. We are basing this on a simple dominant recessive relationship. Your hypothesis would be something along the lines of 3/4 of the seeds will be one color or one shape, and 1/4 will be the other color or the other shape. So this is all based on the phenotype. Or if we're talking about the dihybrid cross, 9/16 will be, 3/16 will be, 3/16 will be, and 1/16 will be. And the way I think about this is, so remember, for this one is based on monohybrid cross. Let's just do a simple one. Again, this isn't the one from the lab because that would be giving you the answers. Right, so if we put those together, we would expect 3/4 to be purple and 1/4 to be white. All right, so then let's do this one. So this is a monohybrid cross. And then this one would be the results of a dihybrid cross. Okay, so example of a dihybrid cross. That would be a dihybrid for the P gene, and let's see, what other one might we want to do? Let's do a dihybrid for the T gene. So a dihybrid individual is going to be big P, little t, times big P, big T, little t. That's a dihybrid cross. All right, so let's look at this individual and let's make some eggs. There's 4 letters there, so I'm going to make 4 eggs. Even if they're all the same, I still just do that. It's easier for me to do. All right, so you can use the FOIL method. Remember the FOIL method? First, outside, inside, last. First, big P, big T. Outside, big P, little t. Inside, little p, big T. And last, little p, little t. That's the FOIL method that you probably learned in middle school or high school. So that takes care of this individual. Now this individual, we're going to have it be spermies. Same thing. First, big P, big T. Outside, big P, little t. Inside, little p, big T. And last, little p, little t. Now we're going to put this on a Punnett square. Boop, Boop. Boop. Boy, is it easier to draw when you actually can see where your hand is on the thing. All right. This exercise is not made for someone who is OCD. It's going to put these eggs over here on this side of the Punnett square. All right, we're going to cross them together. Big P, big P, big T, big T, big P, big P, big T, little t, big P, little p, big T, big T, big P, little p, big T, little t, big P, big P, big T, little t, big P, little t, little t, big P, little p, big T, little t, big P, little t, little t, big P, little p, big T, big T, big P, little p, big T, little t, big P, oh, hold up, ah. There we go. Little p, big T, big T, little p, big T, little t, big P, little p, big T, little t, big P, little p, little t, little t, little t, little p, big T, little t, little p, little t, little t. So that's how we would do that. Right? Keep in mind, each gamete needs to have one copy of each gene. Right? So this one has one P and one T. If we go through there, we can do the phenotypes. I'm going to do pheno. And I'm going to say, okay, double dominant phenotype, there should be 9 of them. All right, dominant P plus recessive T, there should be 3 of those. All right, recessive P plus dominant T, there should be 3 of those. And then we have a double recessive. There should be 1 of those. So you should have a 9 to 3 to 3 to 1 phenotypic ratio. Now, if you look at this, 9 plus 3 plus 3 plus 1 equals 16. That means that 9/16, 3/16, 3/16, and 1/16. That is what we have for the probability that we would have a double dominant versus a dominant P with a recessive T, a recessive P, a dominant T, or a double recessive, if we put those all together. So if we go back here, 9/16 are going to be double dominant, but you'd want to write out what that meant. 3/16 are going to be, number 1 is dominant, I'll put number, plus number 2 recessive. 3/16 are going to be number 1 recessive plus number 2 dominant, and 1/16 is going to be double recessive. So you need to start out with this when you're starting your chi-square analysis. What do I expect? Okay, so I think this is a monohybrid cross. I expect this and this. Oh, I think this is a dihybrid cross. I expect this, 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 and this. You have to have your expectations before you can determine if what you see meets your expectations. And that is also a life lesson. All right. So here is an example. Let's say you and a friend are, I don't know, for some reason we'll say this is a time when you can actually have social get-togethers. You have just had a really nice dinner together, and you're going to flip a quarter to see who has to do the dishes. Fun. So if you flip your quarter 100 times, you would expect 50% of the time you'd have heads and 50% of the time you'd have tails if the quarter is clean and normal and everything goes the way it should. All right, so if you flipped it 100 times, you would expect 50 heads, 50 tails, or a 1-to-1 ratio of heads to tails. Well, there was a study done several years ago that found that all of the money that was surveyed in the United States, which was quite a bit of it, coins and bills, had a little bit of feces and a little bit of cocaine on it. Now, maybe your friend is the one that ends up having to do the dishes, and your friend says, hey, I think that your quarter is either A, rigged, right? It's one you could buy on Amazon. It's a quarter that is weighted, or B, isn't flipping normally. So maybe there's a little bit more feces on that one than you expected. I don't think that your quarter is fair. So you can do a chi-square analysis to determine if your quarter is fair. So you're going to flip your quarter 100 times. What you found was 55 of those times it was heads and 45 of those times it was tails. And your friend says, aha! The quarter is not fair. You say, nah, that's not a big enough difference. Period. That difference is just due to chance. It's fair. Okay, so let's test your hypothesis. Okay, so the first thing we want to do, deviation, observed minus expected. 55 minus 50 is 5. All right, 45 minus 50 is negative 5. Then we're going to square them. This is your deviation squared. 5 squared, 25. Negative 5 squared, also 25. All right, d squared divided by expected. 25 divided by 50, 0.5. 25 divided by 50, 0.5. We add these together. So this is your d squared divided by expected. Your chi-square is the sum of those. 0.5 plus 0.5 is 1. This is your chi-square value. Now, in order to move forward, we need to know the degrees of freedom. Right? Number of categories minus 1. Okay, so our degrees of freedom here, number of categories, 1, 2, minus 1, 1. Degrees of freedom, 1. Now I think my chi-square table's on the next page. It is, yay! Okay, so here we are. It's accepted that this is where we want to look. All right, all of these fit our hypothesis. So the data fits the hypothesis if we're there. So how to read this? We go to degrees of freedom. We said we had 1 degree of freedom. We are going to look in this column. So specifically, if the quarter is fair and that difference is only due to chance, our chi-square value will be somewhere between 3.84 and 0. What was our chi-square value? It was 1. All right, so we are right in here. That tells us that the quarter is fair. It's because it falls between a 0.3 and a 0.4. so the quarter is fair. Okay, so we can say to our friend, haha, that difference between what we expected and what we saw was just due to chance. Hypothesis is supported. Your hypothesis. You have to be clear on which hypothesis you're checking. Right, so here our hypothesis is that the quarter is fair. We would expect a 50-50. We went with that hypothesis instead of our friend's hypothesis because our friend's hypothesis didn't have anything specific about it. So we couldn't come up with an expected number or an expected ratio from the quarter is not fair. But we can come up with an expected number or an expected ratio from that is just difference due to chance, because we would expect a quarter flipped 100 times to be heads about 50% of the time, tails about 50% of the time. All right, so here's another thing to think about. If you flip the coin and the first time you flip it, does that mean, if it's heads the first time, wow, that's terrible handwriting. Does that mean that you would definitely expect it to be tails the second time you flip it? No, it's a new event. So every time you pick up that quarter to flip it, there's a 50% chance that you're going to have heads and a 50% chance you're going to have tails. So the outcome of one flip does not affect the outcome of another. And that's really key. Right, that might seem really straightforward when it comes to quarters, but now think about it with children. So my husband's family has 4 boys and a girl. The first 4 children were boys. The oldest was born. There's a 50% chance that that child was going to be born and be male, and a 50% chance it was going to be female. So when the baby was born, it was male. The second child, same thing. 50% chance it was going to be male, 50% chance it was going to be female. It was male. Third child, same thing. Fourth child, same thing. Every time those parents conceived, there was a 50% chance that that offspring was female and a 50% chance that that offspring was male. Now, this is taking into account only the straight up genetics of sex and gender, which those are not necessarily straight up, because as we talked about in a previous chapter, there are other things, other genes, other chromosomes that influence whether you actually end up physiologically female or male. But we're talking about chromosomal sex here, XY versus XX. So every time a couple conceives, there's a 50% chance you're going to have XX, which would be chromosomally female, and a 50% chance you're going to have XY, which would be chromosomally male. Even though my husband's mom had 4 boys in a row, there was still a 50% chance that that 5th child was going to be a boy. So that is another thing to think about. Every time you do a genetics cross, when you look at the offspring from that cross, it's almost like a new day. So if we look back at the peas, let me flash back to the peas. Boop, 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 boop. Here, right, if we look at this one, the first time we mate these two, we have a 75% chance that it's going to be purple flowers on the offspring, a 25% chance it's going to be white. Next time we mate them, same thing. So if we count 3 purples, that does not mean the next one will be white. So we use our chi-square table and the chi-square analysis just to tell us if those data fit our hypothesis or falsify our hypothesis.